Vanakkam. Welcome to this course on classics in neuroscience. We just started looking at the biology of the cell, or the cell biology and the synapse. In this video, we will be discussing what was the early conception of the synapse, how the term synapse was introduced or who introduced it, and this debate of more than 120-130 years ago, whether there was neurons or neurons were separate structural and physiological entity, or there was connection, there was reticularism. This debate between Ramon y Cajal and others. In the 1890s, Santiago Ramon y Cajal, who is considered as the father of modern neuroscience, proposed the idea that neurons were the smallest functional unit of the nervous system, and neurons communicate with each other through distinct mechanisms. So, it is not like the nervous system is continuous, but rather there were discontinuities between discrete cellular structures or discrete cells called as neurons. This idea came to be called as the neuron doctrine. We will discuss this in greater detail in future classes. This came to be called as the neuron doctrine. For the 1890s, this idea was truly groundbreaking. More importantly, it was not clear how neurons communicated with each other. If each neuron was a distinct discrete cell, how do the neurons communicate with each other? This was not clear. So, there was this proposal or an idea that neurons interact through some sort of contact or interaction between their axons and dendrites. The exact nature of these interactions was not very clear at that time in 1890s, which is why this is a groundbreaking concept. You do not understand how this works, or this was just imagination of things that never were, and ask why not, that kind of question. Why not? This can be a possibility. So, this is more like an imagination or a figment of imagination from Ramon y Cajal. Why not? This is a possibility. Remember, we have been discussing this, why imagination is more powerful than knowledge. But the question still remains, how do neurons communicate with each other? The early suggestions were that electric currents in an inductor coil, how are they induced? Not necessarily through contacts, not necessarily through contacts. Similarly, electric current that flows in one neuron induces an electric current in the neighboring neuron, using some analogy similar to how it is happening in an inductor. This shows how limited knowledge was of the biochemical nature of communication between neurons in the 1890s, early 1900s. Of course, as per our previous discussion, acetylcholine and adrenaline were discovered much later, 1910s, 1920s, 1930s and so on and so forth. Only in the 1940s and 1950s, methodology was available to identify this in a much better manner. The English neurophysiologist Charles Sherrington worked extensively on reflex functions of the spinal cord. He suggested that reflex was the basic functional unit of movements and movement generation, but he wanted to understand how impulses were transferred from neurons of the spinal cord to muscles. It was not clear to him how that happened. So, he wanted to understand how this can happen, that communication between neurons and muscle cells can happen. For example, he suggested that there can be a unique mechanism of how impulses are transferred from one nerve or from one nerve fiber or from one neuron to another neuron that is distinct from how conduction happens within a neuron. So, that is how communication happens between one neuron and another neuron is different from how communication happens from within the same neuron. This is the suggestion that he made. Let me read from his original text. He says that so far as our present knowledge goes, present knowledge is 1897. Okay. So far as our present knowledge goes, we are led to think that the tip of a twig of the arborescence is not continuous, but merely in contact, is not continuous, but merely in contact with the substance of the dendrite or cell body on which it impinges. Such a special connection of one nerve cell with another might be called as a synapse. So, he essentially coined the term synapse. Someone asked him later, how did he come up with this name synapse? One of his students, I think, asked him, how did you come up with the term synapse? So, he wrote back to that student and explained what happened. In 1897, Michael Foster approached Charles Sherrington to author the neurophysiology part of Foster's textbook on physiology. So, Michael Foster's textbook on physiology was the standard textbook of physiology available in the 1890s. Nowadays, we have this Guyton, for example, Guyton and Hall, for example. There are others. So, in the 1890s, Michael Foster's textbook was standard, and Michael Foster approached Charles Sherrington to be the author or to contribute towards the neurophysiology chapter of the physiology text, because Charles Sherrington was this new, young and up and coming physiologist, neurophysiologist of the time. So, he considered many names, and he was not sure how to this junction, how to describe this junction between neuron and neuron, neuron and neuron junction. Sherrington originally suggested the name syndesm. This was the original name suggested by Sherrington. Michael Foster contacted his friend from Trinity College, Verrall, and Verrall suggested that maybe synapse might be a better name. The term synapse essentially describes the specialized junctions between excitable cells, neurons and neurons, neurons and muscle fibers. This marks an crucial moment, an extraordinarily significant or important moment in the history of neuroscience to identify the junction between a neuron and another neuron as synapse. The word synapse itself is derived from Greek synapsis, which means contact or junction. This concept is absolutely critical for understanding of neurological functions. It turns out that even after 100 years or 125 years, it still continues to influence the field of modern neuroscience. How we understand neuroscience depends to a great extent on how neurons communicate with each other through the synapse. Shown here is an example of how one neuron communicates with another neuron through the release of, in this case, through the release of chemical transmitters, neurotransmitters through vesicles. See here, there is a vesicle that is packed, that's packed here, and that's getting released here. This process of release of vesicles is called as exocytosis. That was not understood in the 1890s, that was described much later. We will discuss that in future videos. So, the release of this neurotransmitter from the vesicles is called exocytosis. An example showing how communication happens between neurons. So, in the late 1800s there was this debate between whether neuron was an independent or distinct structural and functional element of the nervous system, or was it continuous. The neuron doctrine was proposed by Santiago Ramon y Cajal and other classical histologists. What they did to support their claim, what evidence did they provide? They essentially stained, they found Golgi stains demonstrating discontinuity between the axon and dendrite. But other people claimed that there is no discontinuity, I am able to see a continuity between neural process, between an axon and a dendrite, I am able to see continuity, I do not know what is it that you are speaking about. So, there was not convincing evidence suggesting that these two neurons are indeed disconnected, they are two separate units and communication was somehow happening between them. So, there was no clear morphological or structural evidence for synapses. This created a lot of skepticism in the 1880s, 1890s. An interesting model to study connections or synapses was the neuromuscular junction or the neuromuscular synapse. This is the synapse between a neuron, a motor neuron and a muscle fiber. This was an excellent model to study. This was first described as early as 1860s and 1870s by Willy Kuhn of Germany. Remember, with the limited stains that Willy Kuhn had access to in the 1860s and 1870s, the rendering of the neuromuscular junction that he provided was not very different from the rendering that later became available. So, for the 1860s and 1870s, the work done by Willy Kuhn was of great significance and essentially constituted great progress. So, Willy Kuhn theorized that the neuronal impulse would propagate from the cell body through the axon of the motor neuron into its terminal, subsequently triggering muscle contraction through some unknown mechanism. We do not know what that mechanism is. Remember, in the previous set of videos we discussed the claim made by Dubois-Raymond. Just make sure the spelling of the name, I think it is R-E-I, yeah. Dubois-Raymond in 1877, I think, where he suggested that the muscle activation might involve stimulatory secretion of a powerful stimulatory substance, This was the claim made by Dubois-Raymond in 1877, but somehow this idea from Willy Kuhn and Dubois-Raymond were not considered so important, or were somehow not given enough significance by those who were studying neuron doctrine, because if they were able to, or if they had considered this work, maybe they could have given this as the possible mechanism of how neurons communicate with the muscle in this case, or with other neurons in other cases. But at that time somehow that communication was not present. Remember, this was the 1880s and 1890s. So, with this we come to the end of this video. In this video we looked at what essentially constituted an early concept of the synapse, and who introduced the term synapse, and how was it introduced, and the history behind this, and the great debate between Neuronism and Reticularism, and how early work on the neuromuscular junction was somehow not given enough importance, or somehow did not shine enough to support the neuron doctrine at that time. With this, we come to the end of this video. Thank you very much for your attention.